Hi guys, welcome. So today's brief video is on module 4, biodiversity, the last part, which is about the evolutionary relationship, comparing the different species using the amino acid sequence, the DNA base sequence, and so on. So let's start. I'm going to start off with this just to give you an idea. Look at this picture. I'm giving you like different organisms, snakes, lizard, even a worm. So in terms of evolutionary relationship, I mean, you don't have to believe in it. Just for the exam purpose wise, we could say that all of them, they have a close relationship because they look similar, right? Now, that was before scientists used to think, oh look guys, they look similar, so they have to be related in terms of evolution wise, they must share some common ancestors. But now we can go a bit more in detail. So this is the advances in technology. Now there are like 4 marks for this. So the first mark would be that, look, I can compare the DNA base sequence of one organism and compare it with another organism, and if they're both similar, then that tells me how much related they are. So the more similarity, the more closely related they are. So let's have an example like humans and chimps. They share 94% of DNA base sequence similarity. That I guess might indicate, or you may argue otherwise, that maybe we are closely related. You also have humans and lions, 95%, humans and chickens, 60%, and so on. But for exam purpose, we will say that if the DNA base sequence of one organism is like similar to the another organism, then that tells us that they could be related. If they are really, really similar, then they are more related. When we compare the DNA base sequence, we are comparing the genes. So you want to make sure that you have picked up a specific gene. So let's say in a human being, you will pick the insulin gene and see the sequence, and then you'll go to a chicken and get the chicken's insulin gene and check that sequence and see if both sequences are they similar or are they different. It has to be gene to gene. Hopefully you get that point. Now, for the exam marks, remember AQA, crazy about all keywords. Make sure, guys, you put down DNA base sequences. Don't miss any of this word out. DNA base sequences. It's all about sequence. That is going to be the most helpful thing. Let's say let's talk about that first one, the first thing, insulin gene, right? Let's say you can't get access to the DNA base sequence. What can we do then? What else can we get? Bingo, mRNA. Insulin gene will be made into transcription, then translation, then to make insulin protein. Let's get the mRNA sequence. So the mRNA sequence, as we know, is a transcription, is a copy, it's like a reflection of the DNA base sequence. So get my mRNA sequence of insulin and compare with the chicken's mRNA base sequence. Then same point actually, the more similarity they have, the more closely related. So keyword here, mRNA base sequence. Very simple. Another mark. Let's say we don't have the access to DNA base sequence, we don't have access to mRNA base sequence. We're talking about proteins. So what else can we do? It's the primary structure of protein, is the sequence of amino acids. So I'm going to get my insulin protein, the primary structure of the polypeptide, and obviously my insulin is going to have a particular sequence of amino acid, right? So remember, we have 20 amino acids and they will be in a uniquely ordered fashion, in a unique sequence. Who determines that sequence? The DNA. Okay, so I'm going to compare my sequence of amino acid with the chicken sequence of amino acid of the same protein. If the sequence of my primary structure of amino acid is the same as the chicken, then if they're really similar, then that tells us that we probably are closely related and we have some sort of common ancestor and so on. So keyword here, amino acid sequence. Don't put the word base here. The base only goes for mRNA and DNA. A lot of students put base for some reason. They just get confused. Make sure, guys, you know what you're talking about. Amino acid sequence. Now, shall we do the last one? Let's think. It's quite difficult. Or shall I make a separate video? You know what? Let's go for it. Last one, guys, is bloody hard. It's not hard because I'm going to explain it. It's hard for normal students because probably poor explanation or they're not really focusing. And guess what? Only like 1%. No, I'm being too generous. Less than 1% of the whole students of the whole country nationally, examiners report said that they can't even articulate this concept. Let alone get the mark. They can't even write it down. What are the words to put down? So let's go through this, okay? So look, if you get this correct, you get 4 to 3 marks. Easy marks, and that will put you in the high band to get those A grade, because most of your peers will not get this correct. So now we want to compare me versus the chicken. Are we similar? Let's say you can't get access to DNA based sequence, mRNA based sequence, you can't get access to that, and you can't get access to the amino acid sequence of the primary structure of the protein. But we can now use the protein itself. The structure, is it the structure? Yeah, how the protein is. You know the proteins, outside the proteins, they have these antigens. We can compare the antigen of the insulin and the antigen. Let's not use insulin, it's probably a bad example. Let's use another protein, albumin. So I'm going to compare myself with a chicken. Okay, are we both similar or not? For this time I'm going to use albumin. Albumin is a protein in your blood, in your serum, floating around. So I want to get my albumin, and I want to see whether the albumin that I have is similar to the chicken. And if it is similar, that means we are related, closely related. If it's very, very different, then thankfully we are not closely related. These are the 5 points. You probably won't understand this. So let me use my storyboard. Over here, the story is a bit different, guys. Sorry, it's not chicken here. Well, you can think of this as a chicken. This is a bird. So, shall we say chicken or bird? Let's say chicken. Let's say chicken. Fine. This is me there, and that's the chicken here, which looks like a bird. The question is, are we related? I'm going to use immunological method. I'm going to use proteins. So the protein that I'm going to use is albumin protein. Okay, so first thing what I'll do is, before we do that, let's go here. This is the albumin protein. You see the outside albumin protein, you have these antigens. These antigens are very, very unique and specific. That's the word, specific. The shape is very specific. Now, antibodies will bind to them if the body thinks it's foreign. The antibody is very specific to this antigen. That's the basic idea before you go and understand this. So first of all, we are going to inject the human... Oh, let me go back. From this person, this is me, I'm going to extract the albumin protein. That's the albumin. It has antigen, unique antigen. And then, what's the word? Unique or specific? AQA examiners, they love to use the word specific. So you have specific antigen. You inject this human albumin into an animal. It could be any animal. Let's say rabbit. So you inject my albumin into a rabbit. What will happen? The rabbit, if you didn't know, has an immune system. Every animal has an immune system. The white blood cell, the lymphocyte, is going to look at the albumin and look at the antigen and say, yo, you look a bit dodgy. The antigen, the lymphocyte, the white blood cell does not recognize this antigen. It does not recognize it, so it's foreign. What does it do? It mounts the immune response. What does it make? It makes lots of antibodies to attack and to destroy that albumin, because the antigen is foreign. So the animal, the rabbit, is going to make antibodies. But these antibodies, well, who will they bind to? It's going to bind to the specific antigens of the albumin. Happy with that? The only reason why we used an animal is to get that antibody which is specific for the albumin antigen. Now we extract the antibody. Okay, happy with that? Good. Then we will do testing. What's our test? I want to see if I am similar, closely related to this chicken. What I do, I get a test tube. In the test tube, I'm going to add in the albumin of that chicken. The albumin of the chicken is going to have its own antigen. And then I'm going to add in the antibody that was made in the animal in response to my albumin's antigen. And that albumin is very specific. Will only bind to antigens that are very specific to that albumin. So then I want to mix them, and then what I should see is I should see the antibody is going to bind to that albumin's antigen of the chicken because they have a similar shape. If they bind a lot, then that means the shape is very, very similar. That means the antigen is very, very similar to my antigen. That's why it's binding a lot. If it does not bind as much, that means the antigen of the albumin of chicken is very different to my antigen. Hopefully you got that point. Now when they bind, the test tube, it causes precipitate. Precipitate literally means a solid mass. It forms a solid mass. So if it binds a lot, it forms more solid mass, more precipitate. If it binds very less, it forms less solid mass, less precipitate. Okay? So if it binds a lot, it causes more precipitate. That's telling me that me and the chicken, unfortunately, are closely related. Because why? Because the antibody is binding to the chicken's antigen, which means the antigen of the chicken is very similar to my antigen, which is not good news. So going back to our 5 points. Pause the video, guys. Write these 5 points, understand it, make sure you know the keywords, and be among those 1% of students who get those 4 marks.